morning students. Today we are going to discuss about embryonic induction. Embryonic induction, it is the process by which one kind of embryonic cell or tissue causes the differentiation of another embryonic part. So here, one kind of embryonic cell or tissue causes the differentiation of another embryonic part. And the tissue which transmits such a inductive influence is known as inductor. Substance that is transmitted by inductor is called evocator. And tissue on which evocator acts is known as responsive cell. So in embryonic induction, one kind of embryonic cell or tissue causes the differentiation of another embryonic part. And this process is known as embryonic induction. And the tissue which transmit such an inductive influence is known as inductor. Substance that is transmitted by the inductor is called evocator. And tissue on which evocator acts is known as responsive cell. Embryonic induction is very important in the normal development of embryo. It has crucial role in cell differentiation. Then it has significant influence on the protein synthetic activity of the responsive cell, in that new cells with new proteins and new biological properties are formed. So due to embryonic induction, new cells are formed with new proteins and new biological properties. There are two types of embryonic induction. One is endogenous embryonic induction and another one is exogenous embryonic induction. In endogenous embryonic induction, here induction is caused by the internal evocators or evocators are present inside the responsive cell. In endogenous embryonic induction, evocators are present inside the responsive cell and cells undergo self-transformation or self-differentiation. It is seen in small size yolk-laden cells or dorsal lip of amphibian blastopore. So in endogenous embryonic induction, cells undergo self-transformation or self-differentiation because here the evocator is present inside the responsive cell. Exogenous embryonic induction. Here, induction is initiated by external evocators such as neighboring cells. It is a kind of contact induction in which the inductor gains contact with the neighboring cells and release evocator. So, exogenous embryonic induction is a contact induction. It's a kind of contact induction in which the inductor gains contact with the neighboring cells and release evocator. And evocator causes self-differentiation. Next is organizer. Organizer is the part of the embryo capable of inducing undifferentiated cells to undergo self-differentiation, diversification and organization. So, organizer is the part of the embryo and it is capable of inducing undifferentiated cells of the embryo to undergo self-differentiation or diversification and organization. Sometimes cells that are induced by primary organizer will again act as organizer. Such organizers are known as secondary organizer. These cells will induce other neighboring cells for differentiation. Then they are known as tertiary organizer. Cells that are induced by the secondary organizer will induce other neighboring cells for differentiation. Then these cells are known as tertiary organizers and so on. Let us try to understand this concept with one example. So we are talking about primary, secondary and tertiary organizers. In the development of amphibians, there are varying levels of embryonic induction. The coda mesoderm, which is seen in the gastrula, after moving inward, induces the overlying ectoderm to develop into neural tube. So in amphibian development, the coda mesoderm, after moving inward, it induces the overlying ectoderm to develop into neural tube. This is the first stage in the chain of embryonic induction. Hence, coda mesoderm is said to be the primary inductor. The coda mesoderm, it induces the overlying ectoderm to develop into neural tube. So, coda mesoderm is said to be the primary inductor because this is the first stage in the chain of embryonic induction. The neural tube then induces the formation of organs like eye, ear, etc. The rudiments of the eye develop as two outgrowth of the prosencephalon known as optic vesicles. The optic vesicle induces the head ectoderm to develop into lens placard, which differentiate into lens. Since the optic vesicle induces the overlying ectoderm to form the lens, it is called secondary inductor. The optic vesicle, it induces the head ectoderm to develop into lens placard and later it differentiate into lens. Since the optic vesicle induces the overlying ectoderm to form the lens, the optic vesicles are referred as secondary inductor. The lens now induces the outer ectoderm to differentiate into transparent cornea. The lens is therefore called the inductor of the third level or tertiary inductor.